Okay, hi everyone. Today we are going to shift gears. We've spent the last couple lectures talking about hypersensitivities as they relate to allergies. And hopefully over the course of the last couple lectures, you've come to appreciate how allergies represent our immune system making a mistake, right? Normally, our immune system should be sensitive and well calibrated to be able to properly detect when things are really a threat or not. Right? So that's why we said, as part of a normal healthy immune system, the calibration or sensitivity of your immune system allows you to not worry about having strong immune reactions to things like different antigens that are in your food, because every day we eat all kinds of different foods that have all kinds of different potential antigenic molecules in them. And it'd be a real disaster, right, if our immune system always freaked out and reacted to different types of antigenic peptides and things that are being absorbed into our GI tract. Likewise, every single day we're surrounded by all kinds of different molecules in the air, on surfaces that we might breathe in or touch, that also would certainly be foreign looking antigens to our immune system. And so thankfully, a properly calibrated immune system allows us to exist in our daily lives and be exposed to things without our immune system freaking out and reacting to everything. We said one of the keys to understand is that our immune system has to actually see that there is some evidence of real infection going on in order for it to activate and respond to something that it sees for the first time. We also discussed that as part of our modern lifestyles, perhaps the fact that we are being cleaner and cleaner and have sanitation, healthy foods, strong medicines to treat different types of exposures and possible infections makes our immune system progressively less well trained, right? Less experienced in having to properly recognize what is a threat and what isn't a threat, because perhaps as kids, with every generation, our kids are cleaner, they're not as exposed to things. And so during the first few years of their lives, into their teenage years, maybe the way that Mother Nature expected us to be exposed to many things like pathogens in our environment, if we deny the developing immune system those opportunities to learn and be challenged and have to respond, right? That perhaps what we're seeing is that every generation of kids, subsequent generation, the kids are becoming more and more allergic, because in the absence of exposure, our immune system adjusts, right? A part of our normal way of our immune system adapting to changing environments. I said if you move to a different location and you're exposed to new antigens in your environment, your immune system adjusts over time so that it knows what is okay and what is not okay and what is worth reacting against. So it may be that what happens is untrained or under-exercised immune systems then are constantly trying to recalibrate, and they get more and more sensitive in a way to try to see if perhaps it can detect anything at all. And then what it ends up detecting are common everyday types of antigens, things like pollen from flowers, or the dander from pets, or different types of antigens in foods, like peanuts, for example, right? And when that happens, the immune system makes a mistake, right? It tries, because it wants so badly to react against something, because that's part of the normal, how Mother Nature sort of expected us to constantly be dealing with things like parasites and other types of microbes in our environment, that then the immune system becomes fooled and makes mistakes and ends up selecting for things like immunoglobulins, for T cell receptors on B cells and T cells that recognize these antigens, and the immune system treating them as a threat ends up building up immune memory, right? So that the next time or somewhere down the line, that same flower pollen or petting a fuzzy animal or eating a peanut, right, causes a strong secondary immune response that can be anything from mildly irritating, right, all the way to being deathly serious. Okay, so let's shift gears a little bit and look at how some of those same principles apply to challenges that are faced in modern medicine when we are dealing with tissue transplantations. So today, tomorrow, and Friday, we'll examine different examples of how the immune system gets in the way of successfully doing tissue transplantations. So to start off with, just a little bit of background thinking about history here. For thousands of years, people have attempted to do transplantations, whether blood or solid tissues. But I think the vast majority of times, it wouldn't be a surprise to you that it was met with failure. Right? So, for example, if you look at this illustration down here on the left, this is an old woodcutting illustration that kind of depicts an early attempt at blood transfusion. So this is maybe around the 1800s, well before doctors had any real understanding of things like blood types. So at the time, and I think through history predating this, people were aware, right, that there is something about the blood that makes it essential for life, not only in us, but also in other animals around us. And I think all of you can imagine that blood was always given sort of this almost mythical kind of quality to it, right? There's something magic about blood and the idea of passing blood from one generation to the next through families, right? That is a very antiquated notion that's been around since the beginning of civilization, right? So it makes sense that for certain illnesses that physicians considered to be a defect of the blood, for example, back when they had an understanding of things like microbes and pathogens, often it was thought that if somebody falls ill, that it has something to do with their blood going bad, right? Or even maybe thinking that they're bad spirits somehow. So throughout recorded history, there are attempts to replace the blood in one person with the blood from somebody else. And not surprisingly, right, oftentimes it involved a family member or somebody who was involved personally with that other person. So here's woodcutting that shows this is a husband donating blood to his ailing wife, right? And of course, the notion at the time was that, well, because, this is husband and wife and they love each other, that somehow this is the best blood to donate. And sadly, even though I don't know the outcome of this particular illustration here, I think sadly, most of the time, right, it was met with severe failure. If you fast forward all the way up until the mid 1950s, it was only then that we actually had the first successful organ transplant involving solid tissue, in this case, a kidney transplant. And it was performed by a Dr. Joseph Murray in Boston, Massachusetts, at Brigham and Women's Hospital. All right, so Brigham and Women's Hospital actually was the hospital that I was born at when my parents were living in Boston. But that was the first time a solid organ transplant was successfully performed, right? Now, I know 1954 sounds like a long time ago because look at this photo, this old black and white photo. But you actually think about it, it's not that long ago, right? We're basically talking about 70 years ago, something like that, right? So really, in the history of humankind, right, that's not that long ago that the first solid tissue transplant took place. Right? So what I want you to think about is that I think all of you are aware that when you're transplanting tissue, whether it be blood, right? Oh, and I think that's very important to point out, blood is absolutely technically a tissue, right? Because I know when you hear the word tissue, you think about solid tissue, right? But the definition of tissue is a population of cells, right, that are functioning, surviving in some sort of a matrix. So that matrix could be liquid. So in the case of blood, hey, we're talking about all the different cellular components of our blood existing in a matrix of fluid, mostly water, plasma, serum, right? So it very much fits the definition of a tissue. So the rejection of tissues may be due to things like blood type incompatibility, which we will examine in depth today, or it may have to do with solid tissue incompatibility, right? For other reasons that go beyond simply not matching blood type. All right, so those two things we'll explore in depth. And I think you'll find it quite fascinating when it makes you think about not only what modern medicine, what sort of challenges we still face in terms of doing tissue transplantations, but also, right, why it's so hard for us to successfully transplant tissues. And that all involves our immune system. Let's start off with blood types, right? Because I think it's obvious to start by discussing the role of ABO blood groups and how our immune system reacts to them and determines things like proper matching of blood types from donor to recipient. So the ABO blood groups, they represent certain types of antigens that are present on the surface of certain types of blood-related cells in our body. Right? Most obviously, and I think what all of you think about right away, these are antigenic signals present on the surfaces of our red blood cells. Right? Our red blood cells are by far the most abundant single type of cell in the human body. Our human body is made of 220 roughly different types of cells, right, that make up a human being. Those 220 or so different cell types, right, end up amounting to several tens of trillions of cells that actually make up your whole body. It's been estimated that in a full-grown adult human, there might be something like on the order of 37 trillion cells that make up the body. Right? Out of those, a good 20 trillion of them are red blood cells, right? So that gives you an idea of how abundant red blood cells are in your body. Right? And so it's not surprising then that antigenic signals on the surfaces of red blood cells can pose a very strong immunogenic type of signal for an immune system to notice or focus on. A surface of a red blood cell has many different types of proteins on it, and many of those proteins could potentially serve as an antigen. But the most important in terms of discussing blood type matching are the ABO blood group proteins and also the RH factor protein. Right? So let's start with ABO. What are these? A, B, and O are different variations, different shapes of a specific surface glycolipid. Okay? Let's look at this cartoon at the bottom here. So here we have some red blood cells, and we're zooming in on these red blood cells over here to take a look at a section of the membrane. So red blood cells membrane, just like the other membranes of cells in your body, is a phospholipid bilayer, right? So you have the phospholipid bilayer in which you have all kinds of proteins and other types of biological molecules embedded. So here you can see, here's this large sort of bluish blob is a protein. That protein, it's made up of many amino acids. So certain amino acids as part of the folded structure of the protein are hydrophobic. So as a result, they naturally are very happy, sort of stuck right in the phospholipid bilayer. Other parts of the protein are hydrophilic. The amino acids are hydrophilic. That means they play very nicely with water, right? So those parts of the protein happily stick out above and down below the surface of the cell, either into the space outside of the cell or into the cytoplasm within, right? Both of which, those spaces are filled with water. Many of these proteins are what we call glycoproteins, right? So glycoproteins have little sugar groups added to them. So that's what these sort of pink, sort of coral-looking branching things in this cartoon are, right? Each one of these little dots would be an individual sugar molecule, and you have them chained together, right, to form some sort of a little oligosaccharide chain sticking out. Right. So you have glycoproteins. You also have glycolipids. In other words, here's a chain of sugars, and the bottom part is actually some sort of a lipid molecule. So the lipid molecule stays nicely in the phospholipid bilayer of the cell, but you'd have these branching sugars sticking out. So the ABO blood types are these glycolipids. So if you look at this cartoon here, right, they have a ceramide group, which is technically a sphingosine with with a fatty acid attached to it, and you have a chain of sugars sticking out. You'll see that these are individual rings of things like glucose or galactose, or a galactose that has an acetyl group on it. So it says N-acetylgalactose, right? And you have a different type of ring, this fucose. So if you have this particular chain of sugars on this sphingolipid lipid group, right, this is known as the O antigen. Right? If you have additional sugars on it, right, the shape of this sugar chain is going to change a little bit. Right? And remember, if we're talking about antigens or things that can serve as antigens for, let's say, an immunoglobulin to bind to, it's all about the three-dimensional shape. And any variations in that shape, right, might make it so that a particular immunoglobulin or antibody can bind to it or cannot bind to it. Right? So all of the red blood cells in your body, as they're being made in your bone marrow, start off with this O antigen all over their surface. So some people have a particular allele in a transferase enzyme, right? So there's a gene somewhere that codes for a protein that functions as an enzyme that transfers sugars from one thing to another thing. And this gene is highly polymorphic. Well, I shouldn't say highly polymorphic. It is polymorphic, right, in the human population. So you might have people who have a version of this enzyme then that will add an N-acetylated galactose group to the end of the type O group. Right? And what would you get? So here you have, over here is type A antigen. So you can see the type O antigen starts off as the basis for this. But if those people have this particular version of the enzyme that can bind to N-acetylated galactose and actually carry it and also bind to the end of this sugar chain, right, it might transfer this N-acetyl galactose to it, in which case the shape now is different than the regular type O antigen. This is the type A antigen. Some people would have a variation in the genetic sequence of this sugar transferase gene so that the enzyme that's made, their version of it, binds to just plain old galactose. And it can successfully transfer that galactose to the end of the sugar chain. Right? This shape is going to be slightly different than this shape. Right? So this is our B antigen. Now, some people are heterozygous. They get one copy, well, everybody gets one copy of this gene from mom and one from dad, right? So if the one you got from mom is the one that makes the A antigen and the one you get from dad is the one that can make the B antigen, then guess what? Your red blood cells, right, are covered in a mix, right? A single red blood cell would have lots of A antigen on it, as well as lots of B antigen on it, right? So you'd be type AB. And then there are a group of people whose variation, allelic variation in this enzyme, makes it so that it cannot transfer a sugar group to the type O antigen, and therefore all of the antigen on the surface of their body just remains as type O. But this is interesting to think about, right? Because as I said before, all the red blood cells that are being made by your bone marrow, right, whether you're type A, type B, or AB, start off as type O, and hopefully you can see why now, right? Because that original type O antigen is the starting glycolipid, and it's only if you have these other enzymes that are able to go ahead and add different groups to it do you end up with type A or type B. So if you just think about then that these different shapes are abundant on the surface of a red blood cell, and red blood cells themselves are so crazy abundant in your body, right? Hopefully you realize, yeah, if you're talking about a different shape, right, that can act as an antigen for an immune system to detect, and there's just so many examples or so many instances of these antigens in your body, it's not surprising then, right, that ABO blood types become a very strong immunogenic consideration. So when you're trying to match donors with recipients, it's very important that you get the typing correct. So here across the top are 4 potential donors, right? Donor who's type O, A, B, and AB. And then on the column on the left are the recipient that you're talking about, the person who needs a transfusion of blood. So I think this is very easy to follow, right? That you need to be able to match up the blood types, but there's a little wiggle room. Why? Right? Type O donors, right, only have type O antigen on their surface. That means anybody could be the recipient and receive this because everybody already, their bodies tolerate type O blood because that's what your blood cells start off from anyhow. That means what? Your immune system, as you are selecting for things like brand new B cells in your bone marrow or developing T cells in your thymus, right? Being able to see type O antigen is a normal part of self-tolerance training. Right? That means no antibodies will be allowed to go, or no B cells that make immunoglobulins that bind to type O should be allowed, right, to go into your circulation in the first place. They would fail the central tolerance training, right? Likewise, any T cell receptors that end up making the right shape to be able to bind to something, you know, that might be similar to this, right? They would not be allowed to work as well. But really, for mismatched blood type, we're talking about antibodies from B cells, right? Now, why would it though? Why would it be that just because, you know, let's say your body, your type O blood, that somehow you would for sure develop antibodies against a type A or type B antigen if it's introduced into you? Part of the reason is because, remember, these antigens are glycolipids, right? So it's like a fat group with a little chain of sugars. So they look very similar to lipopolysaccharides. Right? And those lipopolysaccharides, or LPS, we've learned about in this class. That is the thick shag carpeting that's on the outside of many bacteria. I mean, we learned about this at the very beginning of the semester when we said that that shag carpeting, that heavy sort of forest of lipopolysaccharide molecules, can help conceal different types of antigenic signals on the surface of the bacteria, making it harder for things like macrophages to be able to successfully grab on and phagocytose and destroy them. That's why things like complement proteins are so important, right? Because complement proteins can go ahead and label these lipopolysaccharide coats of bacteria in order to get things like the macrophages to stick better. Right? So because these blood type antigens, A, B, and O, are themselves, right, glycolipids, that means over evolutionary history, our immune system has evolved to be really good at developing antibodies to bind things like lipopolysaccharides or foreign-looking glycolipids, right? So already our immune system is sensitized to choosing for antibodies that can bind them. So that means if you are type O, it is almost a given that over the course of your lifetime, you've been exposed to bacteria and things like that, that you will have already built up certain antibodies in your body that can accidentally bind to other blood types, like the type A antigen or the type B antigen, right? So that's why type O recipients, right, they can only receive blood donated from a type O donor, because it is almost a given that the type O patient's body will already have anti-A and anti-B antibodies present, right, because of the similarity between these A and B antigens compared to the lipopolysaccharides of bacteria. Right? But this also means that a type A person, right, would not have any antibodies against type O, because that's a given, right? And of course, they wouldn't have in their body any antibodies against type A, right? Because as part of their immunoglobulins being developed on B cells, right? If they are type A, then of course, as part of self-tolerance training, they would have weeded out or eliminated any naive B cells that might make anti-type A antibody, right? But they would most likely have antibodies against type B, right? And that means they could not receive blood from type B donors, nor could they receive blood from AB donors. Right? And of course, the inverse is true. If you're type B, chances are you have anti-A antibodies. If you are AB, then you're sort of set, right, as the universal recipient, because you can tolerate blood donated from all four of the potential donor types. Okay. The other strongly immunogenic antigen that's present on the surfaces of red blood cells are the Rh factor proteins. So what is Rh factor? So again, it is a family of proteins on the surfaces of red blood cells and other sort of blood-related types of cells, like the cells that line your blood vessels. So Rh stands for rhesus, as in rhesus monkey, and it was originally identified decades ago when scientists were trying to understand why it is that blood from a rhesus monkey, when they tried to transfuse it into rabbits, right, it would be rejected. So there are several different Rh family proteins. There's roughly 50 of them, but there are, I think, 4 or 5 that are considered the most sort of important when trying to match recipient to donor in blood transfusion. Among those, so I think these are called like type C, D, E, and type D is the most immunogenic. Right? So this is the one that you actually are referring to when you say whether you are Rh positive or Rh negative. What are these proteins? So Rh proteins, especially RhD in particular, are membrane proteins that function as an ion channel. So here you see the membrane surface here. Here is the Rh protein. You can see starting from the amino terminal end, it is, you can follow the amino acids around and they zigzag back and forth through the membrane, eventually ending at the C-terminal end of the protein. So this is 417 amino acids long. So in real life, of course, in three-dimensional space, these would not just be zigzagging as though they're on a flat computer screen, right? They would actually form a three-dimensional shape. They'd come around and form a little tunnel. And so when that happens, you see these are several of these Rh proteins working together, pink one, green one, blue one, right? They form a channel through which ions can pass in and out of the cell. Right? So for people who are Rh positive, the gene that encodes for the type D Rh factor protein, right, encodes for this RhD protein that then assembles on the in the cell membrane, right? And so you have the presence of this RhD protein. And so when you detect it, you say you're positive for it. But many people, the allele that they carry doesn't create successfully an RhD protein. So as a result, you don't see RhD protein on the surfaces of their cells. So they are Rh negative, or the absence of RhD protein. So you might say, well, that's kind of weird then, because are these Rh negative people at some sort of a disadvantage? No, because these Rh proteins are very redundant, right? So just because you don't have RHD or a functional RHD protein on the surfaces of your cells, you have plenty of other Rh proteins and other ion transport channels to make up for it. But what this does mean, though, is that Rh negative patients who receive Rh positive donor blood, right? Again, because when you donate blood, you're donating a lot of blood, right? So many instances of this foreign antigen being brought into an immune system that has never seen it before. So therefore, that recipient will select now for the selection of B cells with immunoglobulins and antibody production to counteract that. So subsequent transfusions with Rh positive blood can be very dangerous. Right? So if you add in the plus minus factor into this, then our simple chart becomes a little bit more elaborate. Right? So not only do you have to consider whether the ABO blood type matches or is compatible, but you also have to factor in whether the RHD status of the donor's blood matches up with the recipient's blood type, right? So you get a chart that looks like this. And so what is interesting about this chart, not only does it help you kind of think about, yeah, this is how you have to, the only compatible sort of matches here, but if you notice, the frequency of these different blood types, right, so across the top here, is not equal throughout the population, right? It varies. So for example, you can hone in here and see that the most common blood type is O positive, right? O positive, and that accounts for about 37% of the population, followed by A positive, right? 35% or 36% of the population. And then things begin to sort of trail off pretty quickly from there. The next most frequent blood type is B positive at 8.5%. And then things get rarer and rarer from there on down. Right? So then the rarest blood type is AB negative, right? Only 0.6% of the population have that. Now, I should say this is not referring to the entire human population all at once, because the reality is, based upon your racial ethnic background, what part of the world you're from, and your ancestry, the breakdown of these percentages actually varies quite a bit. I don't think there's any cases where something is completely flipped around, right? But there is variation in different populations, which makes you wonder, because to this day, we still don't know the significance of the ABO blood types in terms of why it really matters if you're O or A or B or AB in terms of the normal functioning of your blood, right? But there must be a reason why the percentages vary from population to population. So there is some evidence of different types of protective qualities, right, in epidemiologic studies, that maybe whether you're plus, Rh plus or minus might have some sort of protective type of quality against certain types of diseases. Likewise, whether you're O or A or B might have some protective attributes for fighting certain diseases that maybe are more prevalent in different parts of the world where your ancestors come from, right? And therefore, those genetic variations are carried in your ancestry and in your genome. But I also want you to think about this. Right? All of this stuff that we're talking about here, like making a chart like this or a table so that you can properly match donors with recipients, right? Where does Mother Nature factor into this? And hopefully you realize, right, this whole exercise of matching blood donor and blood recipient is something that's actually very unnatural. Right? Mother Nature never intended for your body to receive a lot of volume of somebody else's blood, right? I mean, on the one hand, I guess that seems kind of obvious, but then on the other hand, wait, Yeah, it's kind of weird if you think about it, right? I mean, look, if you get into a fist fight, you throw out, we're just animals, right? So, at the history of human beings, we fought with each other. Yeah, and you might end up getting cut, you bleed, and you get a little bit of your adversary's blood mixed in with your blood, but that's a tiny amount, right? In the case of mothers giving birth to newborns, right? We stressed that normally mom's blood and baby's blood don't mix, but during delivery, when things get kind of messy, right, there is a very strong likelihood that there might be a little bit of mixing between fetal blood and mom's blood. But again, that's a pretty small volume. If, on the other hand, we're talking about doing a blood transplant, right, we are talking potentially like liters of blood, right? And that is not something that Mother Nature designed us with an immune system to be able to tolerate. That's why matching the blood type is so important. And it's entirely an exercise that human beings have come up with ourselves, right, in order to save somebody's life, to transfuse blood. All right. So last week we were talking about how allergies are mistakes that our immune system makes when it is faced with something that in reality isn't a threat, right? But it ends up being interpreted as a threat. And so we said that's why allergies are based on this concept of hypersensitivities. So let's continue this, right? Because guess what? These types of immune reactions that happen with your immune system detecting somebody else's blood type, that is also your immune system making a mistake, right? If you are the recipient of a blood transfusion, you want your immune system to tolerate and accept that other blood, that donor's blood in your body, right? Because that's what we're trying to do. Again, trying to fool Mother Nature here, right? By replacing somebody's blood with blood from somebody else. But unfortunately, as we've been discussing this lecture, many of those immunogenic antigens on the surfaces of red blood cells, right, are really prone to being detected by immune systems as being something wrong, and therefore blood mismatching is a type 2 hypersensitivity. Remember, type 2 hypersensitivities involve IgGs that are selected for and then built up as part of an immune response to try to fight against something that the immune system sees as a threat. And importantly, that elevated IgG level might stick around as immune memory. So it's the sugars on these AB type glycolipids and or the presence of Rh factor protein on mismatched blood that leads to IgG attack on the donated red blood cells. Right? So what happens is that the patient's body, right, their IgG that have been sort of selected for to detect things like anti-A antigen or anti-B antigen or anti-RHD, they bind to the donated red blood cells. They cause the red blood cells to hemagglutinate, meaning they cross-link and clump together, right? As well as causing the red blood cells to also be attacked by complement proteins. Because remember, IgGs, when they fix onto the surfaces of targets, right, their stems are very good at attracting things like complement proteins that can then also form things like membrane attack complexes, turn the outer membrane of a red blood cell into Swiss cheese, right? And then the red blood cells will lyse. So if this happens on a large scale, because again, we're not talking about you got into a fist fight, so you got a little bit of your adversary's blood mixed in one of your cuts, right? Or a mom giving birth to a child, and there's a little bit of fetal blood mixing with moms. If you're talking about liters of blood going into your system, you better believe, right? There's going to be a strong immune reaction. So this leads to things like fever, chills, renal failure, right? Because suddenly your blood is filling up with clumps of red blood cells that are agglutinated together. And so your kidneys, right, get clogged up with these clumps of red blood cells. Yeah, right? And if untreated, this will lead to death. Right? So fortunately, ABO blood typing is a very straightforward, easy, and inexpensive step in making sure that there's compatibility between donor and patient. I mean, so many of you who are taking immunology lab this semester, you already did this exercise, right? But let's kind of review, right? For those who haven't or are not familiar with it. Typically would have 3 sort of test tubes or maybe little wells on a test plate that you would put serum that contains antibodies against A, B, and also the RHD, one in each tube. So this tube is filled with serum that contains antibodies that bind to the A antigen. The serum in this tube contains antibodies that bind to the B antigen, and maybe in the third one here would be antibodies that bind to the RHD, right? So then into each one of these, you go ahead and you put the unknown blood and you mix it up and see what happens. And so the result might be this, right? So in the A, it looks like the blood nicely diffuses through here, right? And you don't see any evidence of any kind of agglutination. But in the B tube, right, you have agglutination taking place. And so if you were to really zoom in and take a look, you would see that the red blood cells are being cross-linked by the anti-B antibody. Right, so here they're zoomed in even further. Right, so the B antibody is picking up on the B antigen. So based upon this reaction, you would say, well, what ABO blood type is this unknown blood? Well, the anti-A antibodies don't react with it. The anti-B antibodies do. So you would say this is type B blood, right? And depending on what you see in the third tube, you may detect the presence of Rh factor on this blood as well. So maybe the answer would be it is B positive. So we did mention that the blood incompatibilities during pregnancy can lead to this hemolytic anemia of the infant or the newborn. Right? So blood type O mothers can certainly carry type A and type B babies based upon the other allele that baby inherits from dad. Normally, maternal and fetal blood are not mixing, and so the amount of IgG being transferred through the placenta is usually pretty low, right? Because even though mom may already have sort of some anti-A or anti-B antibodies in her blood, up until this time, there's not been much motivation for her immune system to really build up high levels of those, right? And if mom's blood and fetal baby's blood don't really mix, then it'll continue to be that there won't be that much anti-A or anti-B IGs being passed from mom to baby. But the Rh status is more serious. Right? So during delivery, when there is the mixing of a little bit of baby's blood with mom's blood, I mean, I think it's inevitable, right? Because childbirth is a very messy event, and you have tissues that are actually being torn up, right? So you have the placenta tearing off the endometrial linings. There's definitely going to be the rupture of many blood vessels and the mixing of blood. So that means exposure. If mom is Rh negative, but during childbirth, she's exposed to some of her baby's Rh positive blood, again, because baby became Rh positive because he or she inherited that from dad's side. Right? As we learned previously, that first delivery of the child, no problem, right? But in the intervening years after that, mom's immune system might very likely start learning to build up tolerance, or not build up tolerance, build up sensitivity to the Rh factor, so that the next time mom becomes pregnant, right, with the couple's second child, right, if that second child, while still in the womb, is Rh positive, then it could be a problem, right? Because then moms would have had elevated levels of anti-RHD antibodies, of IgG that would then be transferred into baby when baby is still in the womb. And that could lead to then, right, baby's red blood cells being damaged. So as we discussed last time, one way that we now treat this is that moms are actually given an infusion of high quality anti-RHD antibodies during pregnancy close to childbirth, so that any exposure of mom's immune system to this new antigen, during delivery, right, her naive B cells that she happens to have in her body that day that would form the basis of the selection would be discouraged from being selected. Right. So we call this type of reaction, when you mismatch blood, hyperacute, right? Again, because you're not talking about a little bit of blood. If you're doing a blood transfusion, you're talking about a massive amount of tissue, and it's tissue that's just coated with these strong immunogenic signals, right? So the response that you get is very fast. The endothelial cells that line our blood vessels also have ABO blood groups on them as well. Right? So not only blood transfusions, but now we can kind of start introducing our discussion about solid tissues as well. So let's say you have a healthy kidney here. Okay. So on the operating table, this is a patient who needed a kidney transplant. And so they received on the operating table this healthy kidney, right? So the surgeons transplant the kidney, they attach all the plumbing, like the renal artery, the ureter, whatever, right? But unfortunately, they screwed up. Why? Because they didn't get the ABO blood type matched properly. Right? So you say, well, why does that matter? We're not talking about blood transfusion, right? We're talking about a kidney. Oh, but the tissue of the kidney, think about this, right? Inside the tissue of the kidney, it's crisscrossed with all kinds of little blood vessels, right? All kinds of little capillaries. And those capillaries, so here's a cross section of one, right? On their surface have ABO blood antigens on them. So if you have an ABO mismatch, what happens is right away, the antibodies in the recipient's body, right, as the blood starts flowing through the transplanted kidney, once it's hooked up, these antibodies go to work binding to the inside of all the capillaries that run through the kidney, right? And they start mounting an immune defense. So things like macrophages begin to attack the capillaries, and they start destroying the capillaries. Right? So that means within just a few hours, right, within a few hours, the tissue, the transplanted kidney, may already be damaged beyond repair because all the capillaries inside have been destroyed by the recipient's immune system, right? Because it was the wrong ABO blood type. Fortunately, that doesn't happen that much these days, right? Because ABO blood typing is so simple, you'd really hope they wouldn't screw something like that up. But again, if this happens, it might happen so fast that before the patient is even off of the operating table, right, after he or she's been sort of sutured up or whatever, it might already be too late, right? The immune system has already attacked the kidney and destroyed it. Okay. So the theme that we're going to go on with starting next time, this is the last slide for today, so we'll end with this, right, is I want you to be aware that as we talk about these different ways that immune systems reject tissue transplants, right, these still follow the same types of hypersensitivity reactions that we learned about as the basis of allergies. So in this case, what we're saying is that there are allorecognition problems. Allo comes from Greek, meaning other, as in something that's normal, but it's just something else, right? Meaning an antigen that is totally normal and healthy from a human being, but the problem is it came from some other human being, right? And so they follow type 2, type 3, and type 4 hypersensitivity models. Remember, when we talked about allergies, we said that type 2 involve immunoglobulin IgGs, recognizing different structures that are sort of foreign and don't belong on the cells of the body, what the immune system is not used to seeing. Type 3 involves antibodies forming immunocomplexes with each other, clumping up. And type 4 doesn't involve antibodies, it involves T cells. T cells that see something different and foreign, right, and then are selected for and attack. So that's what we discussed with allergies. So with grafted tissues, they followed these same models. Hyperacute reactions are what we talked about today. So this is reactions that happen very quickly and severely due to ABO mismatches, right? So in this case, in the allergy, we made fun of this cartoon. We said it's like a hot dog and a corn dog, right? But the whole point is this resulting shape is something new that the body didn't recognize, right? So what is this in the case of ABO blood mismatch? Oh, this is the A, B, or RHD protein, right? That the recipient's body never saw before, right? And has antibodies against. Wednesday and Friday, we'll talk about acute reactions and chronic reactions, right, in solid tissue transplant. And that's not due to ABO mismatching. You can get the ABO blood type matching right, when you're about to go ahead and transplant solid tissue, but there are still many other things you have to worry about. And those have to do with HLA or MHC mismatches, right? So that's kind of exciting. We'll get to that. The last thing I want to say is notice type 1 hypersensitivities that you normally see in allergies, right? We said those are the most sort of commonly thought of allergies, like you breathe in pollen or cat dander or eat a peanut that you're not supposed to, and you get a strong, potentially life-threatening response. That's a type 1 hypersensitivity. Why do we not see type 1 hypersensitivities in tissue transplantation scenarios? Why? It's because, remember, type 1 hypersensitivities and allergies involve the production of IgEs, IgEs that have been selected for because you mistakenly saw something like pollen or peanut oil or cat dander as being a threat before. So your body built up IgEs, and now your mast cells are ready to respond, right, to these and give you an allergic reaction. In the case of tissue transplantation, there's no reason why your body would have ever been exposed to somebody else's blood or somebody else's tissue, solid tissues, before this transplantation event, right? So there would be no reason why you'd have IgEs built up. So that's why we don't see type 1 hypersensitivities as part of tissue transplantation, right? Okay, so why don't we go ahead and stop there today. Stop the recording.